the H2O match, and we're going to choose only match if on that as well. So go down to H2O match and tap only match if, please. Thank you. And we're going to just check a small delta on H2O. So just check the box next to delta H2O, please. Thank you. So we have now set it up that the instrument will decide for us if we need to match. What this means is that it's going to match on your first log almost certainly, because we won't have matched yet today. But after that, it's going to look at the criteria for matching. Is the CO2 small? Is the H2O small? We don't need to have the time and reference change checked for H2O. It's not as, it doesn't change as much, but let's just do delta. So if there's not much change in water, if there's not much exchange between the inside of the leaf and the outside of the leaf, then we match. The next thing that we want to do is okay, so the next thing we're going to do is we're going to go down to fluorometer options, please. So the next tab, yep, that one. And here are our fluorometer options. So what we can do is we can say, do we want to actually do a fluorescence flash when we log? We can say no, or we can say we want to do just a flash, or we can say we want to do a flash with a dark pulse. So go ahead and tap that and choose option 1. FOFM for the dark or FSFM prime for the light. And we want to, our options for flash type are, we could choose rectangular, multi-phase, induction, or auto. Let's keep it at auto. Why do we choose that? Well, it means that if our plant is really dark adapted, we make sure to do a rectangular flash. And if our plant is light adapted, we make sure to do a multi-phase flash. We could choose one or the other, but the benefit of using the auto option is if we're a little tired and we don't remember to change it, it doesn't hurt us. So we don't want to do multi-phase flashes for dark adapted plants. And for light adapted plants, rectangular flashes might not give us the FM prime that we need. So telling it auto says, you figure it out. Just like for doing match if, we tell it, you figure it out. You, as the machine, think about it and do that for me. So auto is the safest option. What it does do is you have to tell it, what do I consider to be in the dark? What is the threshold for dark adapted or light adapted? And 50 micromoles is usually a pretty safe number. If it's below 50 micromoles, probably there's not a lot of photosynthesis happening. So that's safe. So we've set this up that we will do a fluorescence flash. We will see that in our data. And then the instrument will decide for us what we need to do. The other thing that we need to do is make sure that our environment is set up. So if you go over to the environment tab, please. So one of the things that I did as we were going through this was I sort of set you up for survey measurements already in terms of flow, relative humidity, CO2 control, fan speed. The things that we might change are temperature. In survey measurements, we don't always control temperature because oftentimes it takes battery life and we might not need to do that. So go ahead and turn that off. We will talk tomorrow about why we might, but for the moment, let's turn that off. And then light. We can't experiencing. So if you were going to go out into the greenhouse, you would need to use your ambient PAR sensor, which we just set up, to allow us to find out what the light is. So in survey measurements, it's very important to check your ambient light and then set your instrument to reflect that, to be close to that. If you're concerned about the ambient light and you think, oh, it's 1273, do I need to make sure to set it to 1273? You don't actually. What you could do is set it to, say, 1250, something slightly lower than the value that you were expecting. So the other thing that we need to do is we need to change our color spec. We set up blue to 100%, and that will not make our plant very happy. So let's go ahead and reset that. So tap on color spec, please. Oh, go back to environment tab. And yeah. Let's go back to the light, please.
Light.
Yep. And tap on color spec, please.
What?
Go back to light, please.
Light.
Yep. And tap on the color spec box here in the middle. Right there. Perfect. Okay. And let's press the little X. Right? And set that to little r. And then 90. So just R90.
Yeah. Yeah, that أنا.
And then press done. Okay.
It's 100.
Yeah, it's okay. So you will need to figure out what the set point should be when you're taking your measurements. Shannon went out and got a plant for me, and so we've set our light level to 200. It's kind of a cloudy day out there here, and so I don't think our light level is very high, so we don't want to set it too high, but we would like to see some plant behavior. So I'm going to see if I can, I'm going to share my screen. And you can see that here is the point at which Shannon put the leaf into the chamber, and you can see that we've got a big change in CO2, and that's because we opened the chamber and then we closed it again. And then you can also see that it's increasing a little bit as you look at it. The plant is getting used to the conditions that it's in. I think this is a bean plant. And so beans tend to be very active photosynthesizers. So you can see that it is able to, I'm going to zoom in a little bit by pressing the plus key. Okay. So this gets rid of that big change in assimilation and stomatoconductance from me having my chamber opened and then closed. But what I would be wanting to do is look for these two values to kind of level off a little bit. And then when I was ready, I could log a data point. So I think that a little bit of homework for you. We would like you to take survey measurements either this evening, although it's getting a little bit late for you, or tomorrow before you come to class. We would like you to take some survey measurements.
Since you have only one instrument, it might be good to, amongst yourselves, kind of divide up the time, and maybe you each get 20 minutes or so with the instrument, and you can practice collecting survey measurements. And again, the idea is that you are measuring the leaf and the conditions that the leaf is in outside the chamber. And you're setting up the instrument to mimic those conditions. So when you put the leaf in the chamber, it will stabilize very quickly. Those of us who've done survey measurements a lot have been able to take a measurement in less than a minute. So we're talking very, very fast measurements. I know that Almaz has had experience with the 6400, so this is probably something very similar. But if you've never done gas exchange measurements or fluorescence measurements, this is really the thing that we want you to get out of these survey measurements. Is the trickiest part is figuring out how to set up your chamber environment and choose the leaves appropriately. So one of the things that we want to emphasize is using your external quantum sensor and really measuring the light that the leaf is currently seeing. So if your leaf is at an angle and the sun is over here, using your external quantum sensor and put it at the angle of the leaf and actually measure what is the light intensity that that leaf is seeing. It might not be the same thing as if the leaf was normal to the sun. So you really need to figure out what that light level should be in your chamber and set it. And that way, when you put the leaf in the chamber, it should equilibrate again in seconds. You should see the conditions equilibrate. You should see photosynthesis and conductance equilibrate. So you can log your data point and go to your next leaf. So the whole point of survey measurements is you're collecting lots and lots of data. So you get a lot of data and you can do statistics with that and really see if there are differences between treatments or cultivars, whatever it is you're trying to test with this experiment. Does that make sense? Yes? Okay, I'm seeing a couple of head nods. Any questions before we let you go?
I will put the survey homework on the screen again in case you want to take a picture of it with your phone or something like that. And again, if you don't manage, if you're not able to do the homework exactly as planned, we just, as Shannon says, we just want you to have that experience of clamping onto the leaf, deciding when to log, entering your log prompt, you know, going to the next thing and then closing the data file, you know, so the experience of doing that quick measurement. And then tomorrow we'll talk a little bit more about some of the reasons that I chose the environmental variables that I did and some of the things to keep in mind about when we take measurements and how we take measurements. But this is the homework for today. If you can graph the homework, here is an example of some homework that. So this data was collected in a Lincoln, Nebraska backyard in 2020. This was during the lockdown time here. So we were all measuring the plants in our backyard. And so this is one of my coworkers' backyard. He had a tree species and then 2 bush species that he measured. And he is now plotting the assimilation versus the stomatal conductance over